And for the discussion, the presence of the environmental pollutants damages the structure of the food affecting its normal physiological behavior. The results indicated that the muscle's food structure was impaired, manifested by a reduction in extensibility of lysis damage to the dark filaments and boost the distribution of tissue structure. Titanium dioxide enhances the biological accumulation of PFOA within the muscle, so it increases its biological toxicity to the organism, so it results in the reduced energy acquisition to the bicep state. So energy was diverted to detoxification and other activities. So that decreases the bicep state production in terms of quantity, quality, and leads diameter that are used for the movement and attachment. So the proportion of the illness on the bicep fracture surface increases, which indicates that the elasticity and the origin of the biceps have been affected. The bicep synthesis predominantly involves cross-linking and combination of various proteins adhering to the substrate surface. PFOA can induce alterations in the bicep proteins. And we also speculate that the physiological stress imposed by nanoparticle pollutants and organic pollutants might lessen the individual muscles' process of food contraction, process release, and secretion. PFOA has been shown to induce oxidative stress from the other resource, leading to apoptosis and DNA damage in biological cells. Furthermore, co-exposure to titanium dioxide and PFOA resulted in significant increase in expression of apoptosis related to PSPC. Pollutants exert a direct influence on muscle gene expression at the molecular level, initiating the activation of key genes essential for bicep synthesis and secretion, such as P3, precolancy, and protein. Interruptions in bicep protein synthesis may lead to the degradation of bicep tissues, undermining their mechanical strength. So for the conclusion, as PFOA and titanium dioxide impair biceps performance, affect its defensive capabilities, and the combined exposure of PFOA and titanium dioxide intensifies the potential adverse impacts by reducing biceps performance, manifested as a decrease in the number of the biceps threads, legs, diameter, RSC, and the peritons, all of them. To enhance the predatory survival abilities of muscles under organic pollutant and nanoparticle stress, muscles compensate by regulating genes related to the strength and adhesiveness. The presence of the pollutants impacts the normal predatory physical defense behavior and the stable survival behavior, and works muscles potentially affects the stability of all ecosystems. And that's all for your time.
Thank you for your nice presentation.
Picture of muscles that was sourced from Alami, you haven't credited it. That is subject to copyright. And a suggestion for you.
Oh, this one, yeah. Actually, because so I have to put the pictures from animals. So we should not do that, right? Oh, okay.
You should have.
I will not mind.
Okay. And a suggestion for you, in the graph section or result section, you have just compacted all the graphs in just one slide. You did not have slide limitation. You must have used different slides for different graphs that will be visible for all.
Oh.
That was my suggestion.
Yeah, actually, thank you.
Thank you. Any more? Thank you. So, thank you, Mr. Kiran, again. Now we moving to next presentation.
That is affected by aquaculture and fisheries. Because of Nepal's topography, erratic rainfall, as well as diverse condition, most of the people of Nepal are economically dependent upon agriculture and fisheries. Also, Nepal's economy heavily relies on natural practices or natural resources as well. Nepal is vulnerable to lots of climate change-induced hazards such as monsoon floods, waterlogging, frost, erratic rainfall, earthquakes. Some of the major risks that most of the communities in Nepal are facing quality livelihoods, food and nutrition security, as well as loss of fish yields due to water stress, pollution of water and land. Most of the people who are getting affected out of climate change are fish farmers, especially smallholders who have really limited technical capacity to adapt and mitigate the climate change effects. And we also know that climate change has disrupted the traditional farming practices. Therefore, it's high time for farmers who are engaging in fisheries to implement climate-smart fish farming techniques that would help them to get away from the hazards. To support and empower women in entrepreneurship and resilience towards climate change, Finnish Agri-Agency for Food and Forest Development have been working with different cooperatives who are involved in fish farming since 2012. So most of the project partners are locally situated in Chitwan and Nomalpanasi. So they are Sundardeep Women Fish Farmer Cooperatives, Federation of Community Forest Users, Amritpur Social Entrepreneur Cooperative Limited, and Finland Foundation as implementing partner. So this project is mainly for enhancing resilience to climate change and disaster risk by developing climate smart production, and its project name is Women for Entrepreneurship and Resilience. So it's basically funded by Ministry of Foreign Affairs of Finland. Under this project, we also generated or created guidelines for climate smart fish farming in Nepal, which is solely targeted to different audience such as fish farmers, fish meat producers, processors, traders, and farming organizations, as well as policymakers and NGOs. So it was prepared by literature analysis, both published and free. We also took lots of interviews and workshops, and most of our major stakeholders were Ministry of Agriculture and Livestock Development, as well as Department of Fishery Development. And fish farmers were included in the preparation of these guidelines were Sundardeep, Papia, and Mishri fish farming cooperatives, because these farmers are locally doing the thing here, so they have a lot of experience and they are also vulnerable to climate change and disasters, so we included them when preparing the guidelines. So you can also go around here. We have stalls and we have presented lots of productions from these cooperatives. So you can see fish feed around, as well as some of our climate smart disaster risk reduction techniques, as well as you can also see climate smart fish farming guidelines, both in English and Nepali, in our stalls. So it was prepared by our independent consultant. His name is Dhruva Gautam, and also, as I mentioned, Sundardeep, Papia, Mishri were the major cooperatives, and Finnish Fish Farmers Association and FFT were the major stakeholders for preparing this guideline. If I have to talk about why this climate smart fish farming is important, then its major objective was to create resilience among the women or among the fish farmers who can get away from climate-induced disasters such as flood, prolonged drought, as well as erratic weather patterns, change in temperature. So when farmers implement these guidelines, they will get away from disease outbreaks, floods, droughts, as I mentioned, as well as these guidelines help farmers to mitigate climate change effects, such as it will help to reduce the greenhouse emission as well. So adapting these farming practices will help farmers to withstand temperature changes, fluctuating water levels, as well as extreme weather events. Overall, this guidelines helps to implement ecological balance and protect the biodiversity, which will help to preserve sustainable fish production. Apart from that, it will also help to improve the food and nutrition security. If all of the fishes that we farm are healthy and they eat nutritious food, then it will also benefit the end consumer, that is we human beings. So overall, it will improve resilience towards climate change actions. Lastly, some of the key actions that we took under this project driven for entrepreneurship and resilience were we helped farmers, especially our fish farmers who are engaged in fisheries, in capacity building workshop and training programs, because these smallholders, farmers are not aware of technical capacity to mitigate the climate change effects. So we help them providing training and aware them about optimizing fish production and increasing efficiency of fish farming practices, water management training, as well as fish disease management training that help them to identify risk of illness associated with fish and how to recognize it. Similarly, we deliver training regarding improving fish seed quality as well as improving fish feed production. So it's presented in guideline as well. If you go through the guidelines, then there you can see, how can we actually help to improve the fish seed quality of fries, fingerlings, as well as the fish feed production. So one of the cooperative mysteries.
I just wanted to ask you whether so this is just a guideline for now, but in the process, in the beginning, have you tried with some method, like the method that you recommend or the method that you say will help reduce greenhouse gas? Do you have any method to measure like that method can really reduce the greenhouse gases or not? And during the making of this guideline process, did you also collaborate with any government agency?
Yes, UNDP and who is into agriculture and fisheries. Actions to Belanasi, we have collaborated with different type of local stakeholders as well. So for installing the early warning system, our Kawasaki municipality government they helped us installing it, and they took the full authority of providing awareness as well as alert to the people and community. So different actions were government definitely collaborating. So and while creating this guideline as well, we collaborated with Nepal Fisheries Association as well.
Okay. Thank you.
Sound is editing sound. So requesting you to present your presentation. So Annabella, please.
Thank you so much. Dhanyavad to the conference organizers, the scientific community, especially Professor Sunil Labrai and Ms. Nita Parajuli for inviting me here. I wanted to talk today about the assessing health of fish stocks through physiology, and especially focusing on temperatures and pollution. So I think we all know that climate change isn't happening. There's no way around that, and that there's temperature anomalies that are happening throughout the world. Here I wanted to show you the map that has everything, the whole globe, and can you hear me better? It's a small room. I'm used to 92 cities, so. But I wanted to show you here the temperature differences. So just for reference, my lab is located right here, just south of Boston, and we are right now here, very red. So you can see that there's higher anomalies for those areas. I wanted to show you a little bit about the system that I worked with. This is Narragansett Bay. It's named after the Native American tribes that lived there. And you can see that this is just south of Boston, and this is opens up to the North Atlantic Ocean. There's two river basins here, and as you can see, and they're very impacted by pollution. So the state water assessment shows high impact areas near the bigger cities. The main challenges that we face are climate change, fisheries bycatch, very strong tourism. All these are beach areas that are very highly occupied during the summer. There's also invasive species. And I wanted to show a little bit of how the land has been changing in the past years. So you can see that in general it's 35% build-up and 39% forest. But throughout the last decades, urbanization has been increasing and deforestation has increased. So we're dealing with a large human footprint and a very high population density that put a lot of stress in the waterways. How are the fish in these communities managed? We have a stakeholder-driven approach. That means that the fishers are taken into account when setting the policies. We rely mostly on permit numbers, fisheries closures, and catch limits. And you can see that it's also done this is based on a trophic level approach. So we look at the whole ecosystem to see if there's any bottom-up or top-down control that needs to be addressed. We have a couple of projects that allows us to do this work, and they're large projects that involve all the universities in the area. So, for example, the project that's just finishing, Coastal Ecology Assessment, Modeling, and Innovation and Modeling. This is actually a view from Providence of the bay. This would be the Atlantic Ocean in the back, and this would be the city up here. And this is a collaboration with the University of Rhode Island, Brown University, Roger Williams University, and all the universities in the state, but also with the state officials through this science and technology plan. We also have a coastal resilience project that's looking at how flooding is affecting the area, and we're really focusing on the community to give us the information that they need so we can build models to predict that. And we're fortunate enough to have investment from the National Science Foundation to increase the capacity in Rhode Island. So basically we're looking at increasing workforce development, so retraining, retooling, etc. We're looking at establishing partnerships within the US but also outside. Focusing on science and communication to be more effective, and also making sure that we can manage the whole projects in a way that all stakeholders are involved. So I wanted to focus a little bit on the physiology, why this physiology project actually matter. And we've been working in the lab a lot on the impacts of turbulence, but also on the impacts of pollutants, such as here is pharmaceuticals. So you can see that pharmaceuticals, this is a work done by Marajuli, that she looked at bluegill sunfishes, adults exposed to estradiol, and they were actually being feminized. And normally we think this only happens in young individuals, but we're actually seeing that adults will actually be able to change throughout their lives. But plastics are also becoming a very important issue and topic in all ecosystems, be it freshwater, coastal, estuary, or marine. We know that plastic ingestion is increasing worldwide, and we know that our beaches and our waterways are getting littered. So I wanted to start talking a little bit about the effects of temperature and some of the work that we've done. We've done this work in 4 estuarine species and 2 freshwater species. So we've worked with 2 flounders, tub, and black sea bass. We also worked with bluegill and largemouth. And what we did here, we wanted to see how the summer temperatures would influence the metabolic rates. So really, we also had a chance to look at their muscles. I won't talk too much about it, but this is the work of 2 undergraduate students and 1 master student that were working with me. I want to show you that I provided the main data from this talk on the abstract, so you can see this graph, you can revisit them later. I wanted to show you here how the temperature is affecting this species. And in blue, you have the two freshwater species, the largemouth bass and the bluegill. This is like a sunfish. And in white, you have the four estuarine species. So two of them are benthic, the scalp and the sea bass are more pelagic species. What we're seeing here is how the metabolic rate compares to the metabolic rate at 20 Celsius. So as you see the darker bars, they're at higher temperatures, and the cooler bars, the blue bars are at colder temperatures. And what you can see is that not all species are the same. So basically, it's very important to focus on the response of each species, especially if you're working in an agriculture system, or if you're actually trying to model fluxes through the ecosystem. We saw that bluegill sunfish was much more affected than the largemouth bass. These animals live in the same waterways. And we also saw that black sea bass and skunk, the pelagic species, had a different strategy from the benthic species. The benthic species actually ended up suppressing their metabolic rates, while the skunk and the black sea bass were increasing it. And the skunk increase is going to be much higher. This is going to lead this species to be displaced. They're going to look for colder waters further north. So this is just what I just told you. We need to be careful of looking at species-specific management and highlight the physiological studies that can feed into those predictive models. We also know that freshwater and estuarine species are both affected, but they're actually affected in different ways depending on their species. We also had a chance to look at acclimation effects. So we had a chance to put fish of two species, skunk and silversides. Silversides are like small bodied fish, and the idea here is to look to see if we acclimate them before testing them, if we're seeing differences in their metabolic rates. So what we did see is that for the basal metabolic rates, acclimation does not have effects. You can see that the small-bodied species has a much higher metabolic rate than the large-bodied species. This makes sense physiologically. We can also see that as we increase temperature, both species will actually increase their metabolic rate as expected. We had a chance to look at maximum metabolic rate. So think of this as the fish has been chased, it has time to use up all the oxygen. And what we were able to see is that now acclimation had an impact, but acclimation only had an impact for the small-bodied fish. So it makes sense, since our acclimation time was only 1 week, that if we were to acclimate for longer than 1 week, we might actually see the same effects on the skull. But acclimation is protective, right? So if we acclimate the fish, or if we do a phase change, they're going to be likely to adapt to their environment, and we can have higher yields if we're talking about agriculture, for example. So this is just to recap what I just told you. Silversides are more susceptible to temperature increases. Acclimation does not affect the basal metabolic rate, and acclimation to warmer temperatures reduces the negative effects, but only on the silversides. So last thing I wanted to talk to you about microplastics. We have a project that's starting this again the Bay, and we're looking at the temporary shifts throughout the year on effluent water. So we predict that because there's higher discharge later in the year with more rains for us in the fall and winter, that will have more microplastics in the system. That's also because even though we keep the water and treat it, if you have a high output, you can have more runoff. And we're trying to do this in a couple of different ways. We want to look at how lentiferous fish and benthic fish, also gelatinous phytoplankton like jellyfish, transport the microplastics through the food web. We also want to do experiments in the lab where we feed known quantities of microplastics and see how that is accumulated. So we know that, for example, oysters are very good at getting rid of microplastics, but fish and other animals have harder time. And we're finding microplastics everywhere around the world, including, for example, they just found microplastics in the blood of newborns. So it's actually affecting human health. So we have to see how it's going up the food web. So far what we're doing is that we're sampling fish in the bay, and we're using a Nile red protocol where we dye the fishes with Nile red, and then we look for fluorescence. We've been able to find microplastics already in the stomachs. So you can see here, this is the stuff that has fishing growth. This is like orange polypropylene microfilament. And we can look at that under the microscope. And here, this is actually microplastics in the silver side intestines. So preliminary work in terms of incidence, so the amount of microplastics that were found in all the samples that we analyzed, it's actually higher in the pelagic species, the skunk and the silver side, by comparison to the other species. These are flounders and little skate. We also looked at two species from freshwater, the species from the freshwater environment, and we didn't find any microplastics. This is not to say that they're not nanoplastics, because we know nanoplastics can actually are harder to detect. But we have some collaborators we can actually look at the nanoplastic in the future. So just to recap, microplastics load on estuarine fishes seem to be higher for freshwater than for freshwater species. And the pelagic fish seem to have more microplastics than the benthic fish so far. So I just wanted to tell you that this doesn't stop here. We have to also make effort to communicate the science to the population, and we do that by doing outreach activities, workforce development, and educational activities. These are just some of the examples of the work we've done, looking at diatoms in the water. You can just go and scrub little rocks, fresh water or salt water with a toothbrush, and then you can have students in the lab just looking at it under a microscope. And we developed this activity where they model it in Lego or Play-Doh or origami to see what they're actually finding. And diatoms can be indicative of water pollution. We're also doing activities where the students develop their own filtration systems in the classroom. And we are deploying the tools and the kits so they can do everything with no cost for school. And we also have some activities that are more towards the heart, how to map what's going on in your waterways. This is two of my students presenting on a sculpture that they designed with artists at Artist Science Competition. And we have a website that's free for everyone to use where you can make simple graphs. This is a great tool for education. So with that, I would like to express my thanks to conference organizers, the Nepalese people for such a warm welcome, and everyone else that I've met at this conference, and the funding sources. And with that, I'll take any questions.
Thank you so much, Dr. Anamela. Effects of increases in temperature and pollution. So Anamela, thank you very much, and the participant. If you have any comment or any suggestion, so we can go for listening to these all. And so please, from the part of audience, especially some postgraduate students I see so far, I guess. So
don't be shy.
So or anybody, especially we like to listen from the young guys. So please first.
But go ahead, Ferdinand. Go ahead.
Anybody Nepalese or Bangladeshi, if you have any question, so please finish, then we'll come to the senior ones. So anybody anything? No? So if you don't ask anything, then I should go to Professor Farooq from Bangladesh Agriculture University. Professor Farooq.
Thank you, sir. It seems I am getting older day by day. Anyway, thank you for your very nice presentation, Annabelle. And I am a bit curious about the temperature map you have shown from 1985 to 2023 that was taken from NOAA, right?
It's the Professor NOAA visualization study, but it actually covers land too.
So it shows the water temperature also? And it is 2 meter high temperature?
It's temperature anomalies, so from the average, the long-term average.
Long-term
Yeah, I'm
average.
gonna get there.
Yes, that one.
Go ahead.
Okay, I was curious about it because there are lots of data provided from several climate modelings, and those data differs from one another, right? About the curiosity, about the authenticity, and so on. Anyway, it was a nice scenario I have seen. But though I am not, my expertise is not fisheries, but I have worked with some of microplastic contamination in several rivers inside Bangladesh. And very unfortunately, we have found a lot of existence of lot of microplastics inside the fishes gastrointestinal tract particularly. And UNEP, United Nations Environmental Program, they predicted that by 2050 there will be more number of plastic bottles inside the Bay of Bengal compared to the number of fishes. So the microplastic contamination is a great concern now. But one thing I want to ask you, did you analyze any FTIR analysis for
So we do have FTIR capacities and ground spectroscopy to actually identify the plastics. We are right now not doing it because we can sometimes for the color or for the properties of the emission spectrum with now reds, we can actually figure out what they are. So we can send some samples to our collaborators and get that FTIR in Rama to actually figure out exactly which plastics. But right now we're just trying to do prevalence, so and just have a bigger picture of what's happening. But you're right. I mean, we are fortunate to be in areas of fresh water that are not very polluted. So I think that we're seeing more in the ocean and the estuaries than we are in the rivers. That's because they're smaller and they're maybe feeding into a local pond, and there's a lot of measures in place to prevent pollution there. Did I answer your question?
Thank you very much. And if the Honorable Chair allows.
That is a great question. We have correlation. Now, I think the correlation will be more at the physiological impact. So we're looking into condition of fishes. So using RNA, DNA to see if the fish are actually decreasing their condition. But there's a possibility that if there's more competition or if there's a shift in the ecosystem, that they'll ingest more plastics because they think it's food. So that is definitely a great point. Thank you
very much. Thank you very much.
Now Professor Salam from the Faculty of Fisheries, Bangladesh Agriculture University.
Thank you for your nice presentation, although I was sleepy. Sorry for that. Water and the ocean water is colder than the land mass, and we can see the higher temperature in the land and the less low temperature in the ocean. And some of the fishes are migratory. They migrate to the land, rivers and streams for feeding or some other purposes. So is there any evidence you have that reduces, because the seawater is colder and the land water is higher, so these differences reduces the migration? And we have our hillside shade that is migrated from sea to river, and due to pollution and other factors, they might gradually less migration occur because pollution, temperature, sound, lots of other things, also the other microplastic as well. So is there any you have done or you know this? It needs huge amount of money to do such research in this way. Thank you.
Thank you for your question. So I think that what we see is a little opposite of that. We're seeing a lot of temperature displacement migration. I think that's what we and so the animals will actually go into the ocean and will be more mobile. And so they're going north. So Black Sea bass, 30 years ago, was not in the bay. It was south for now. And now it is in the bay and it's doing really well. The scub is actually having the opposite effect. They're actually very depressed in terms of their metabolic rate is very affected and they're moving up to the cape. Now we have some barriers because we have a long piece of land and so fish that are smaller won't be able to go through that. So there are definitely considerations to have. Our bays are very, there's a lot of fluctuations in temperature. So what I'm also seeing is that the estuary fish already has some built-in adaptability, right, to changes in salinity and changes in temperature. So they might not be as displaced, but they are being affected. Thank you.
Please identify yourself and ask the question.
I'm MSc part of the graduate as well from Zurban University. So thank you for the presentation. It was really nice. And actually, I wanted to ask you questions on the effect of temperature. And like we can see that the impact, like both negative, positive impact. So in case of cultured species like carps that we have here, a bit of higher temperature to a certain extent is good for us because we feed them and they eat and they grow faster. That's the same with all of the cultured species. But in nature, that's a bit different because we are not aware about the biomass if that really.
So I don't think like in nature we have that biomass or what's your opinion if the temperature is increased, of course the metabolic process will increase and that will cause them to feed a bit high. So what's your opinion on that? Do you think the biomass or the nature can sustain that or do you have any take or any findings on that case?
Right. We are actually working with modelers. We're feeding our caloric rate into their models and we're seeing that actually gives us better predictions of what's available in the bay. But the fish tend to be limited by competition. Be it interspecific competition or intraspecific competition. So food is the limiting resource. So it's not like in agriculture where you can feed them more. We also think that in terms of metabolic processes, it will actually be maxed out. So maybe if you grow carp for several generations, they can actually improve their enzyme faster so that they can actually continue to grow. But what we're seeing is that even with acclimation, most of these fish are not able to expand on that biomass, even if we are feeding them in the lab. So we will have a two-pronged effect. One, these populations are not being bred fast enough to create that adaptability, and the enzymes are not optimized for the temperature, so for their normal processes that we're seeing now when we look at the muscle. So we're looking at the muscle physiology too, and we're seeing that they are not able to compensate for the change in temperature the wild-caught fish might do. At the same time, we don't have enough food, and we're having like problems with the top-down control, right, on the ecosystems and a break in the ecosystems.
Okay, and I also have another question about microplastics. So, from your chart, I can see that now I'm aware. Now I was not aware about microplastics. So, is it fine? Okay.
So now from from your presentation, I'm aware about where in a fish body or muscle the microplastic are located. So from that, from me as a not so expert person, so should I have a conclusion like if I'm eating a fish from such sources where there's a presence of microplastic, should I simply remove the intestine and stomach and that will make the food free from microplastic, or should I worry about if the microplastic might have gone in the muscle as well? From your perspective, I think what I think is okay, so I should remove the intestine and that's good to go.
So that's a great point. So we're seeing that they are higher incidence in the intestine and the stomach, which is good. That means that part of the, especially macroplastics and some of the microplastics are actually being digested. So that's great. But we are also seeing microplastic accumulation in the muscle and in the gills. So we don't eat the gills, but we are seeing it to the muscles, and we're also seeing it to the liver. And it's interesting because when you see it in the liver, it doesn't mean that it's in the liver itself. It could actually be in the blood vessels. And if it's in blood vessels, it's throughout the whole body of the fish, right? So I think we have to be careful. It needs to be monitored. There's other sources, of course. We know that microplastics can be in air pollution, right? You can actually breathe them in, they all go through your mucosas. So there's actually cells in the intestine that have high affinity to microplastics. So we know now that there's a way to transport microplastics into your blood and then allocate it into other tissues. But again, it's good that at least some of it goes out, right? Otherwise, we'll be eating a lot of plastic bottles.
Thank you.
So, I'm a postgraduate student. I have a question. I am from Himachal Science and Technology University. Okay. Does the mouth size of fish affects the consumption amount of microplastic in fish? Does the mouth size of fish? As different fish consume amount is different level. Some fishes level is too high and some level is too low. Why? Is there any influence of mouth?
Right. I cannot tell you directly because we haven't really looked at the effect of mouth size, but I think that a lot of the microplastics that are interested with the food that they eat. So it's like, for example, if I'm a large fish and I ingest a smaller fish that already had that in their guts, that is going to go into my gut. So you saw that small fish that had microplastics in their gut, and the fish, they don't take the guts out, right? They just eat the whole thing. So it's going to go through their systems. It could be that as we go up on the trophic level, we have bioaccumulation, and there's some indication to think that. So more in how higher the trophic level you are, and less maybe on your mouth size. A lot of animals are able to discriminate for plastic for taste, right? In their taste buds, and they might not want it, they might be able to spit it. So we're looking to do lab work to actually figure that out. If they actually are able to spit it, if they detect it, or if they actually will not even notice it. Great question.
Thank you.
Yeah, highest number of questions in this session.
May I supplement a bit?
Yeah, please.
Fish biologist, the main scientist, didn't find out the reason why the whale is dying rapidly. And one sample report, they got the stomach of the whale and they found more than 1,000 plastic bags inside whale's stomach. And what you say, the size
Microplastic and whole plastic and the microplastic, because our concern is with the microplastic that is at micro level and ordinarily you can see it, but it is ingested and maybe contributed in the muscle and throughout the body of the fish. So here the consumption of whole microplastic and whole plastic and the microplastic is coming through the water invisible really, as it is micro. So how do you differentiate this sorts of two ideas? Ingesting the whole body microplastic and the microplastic. How can we separate these two ideas? Because once you consume the plastic, no matter, it will, due to some digestion process, it will be inside the blood and other streams of the body. But microplastic conception is different. That is invisible, but it is impacting the lives and the ecosystems, everything.
Right.
So how do you say that?
Right, and I think that we have a lot of awareness about plastics from sea turtles, from whales, from seabirds, that we see that they can even stop them from. I think that has received a lot of attention. Now, the fish that I work with are small fish. They actually don't eat a lot of plastic bags. They eat fragments. And we're paying the price of the plastic pollution that's been in the ocean for a while. And so we are getting the smaller and smaller plastic sizes because they're overwatering and everything. Larger plastic sizes will accumulate in pelagic environments like the great plastic gyre, or they'll end up at the beach. But you won't find as much plastics in the water, large plastics in the water column in the small estuary. So you have there, our concern is mostly the microplastics. So those are really great questions. It's like, I think that working with plastics is easier because you see them. If you go down and as more time passes, we have to think about the ones that will actually impact physiological processes and that won't actually bioaccumulate. Because again, the plastics in the stomach of a fish that we eat might be okay because we can take the stomach out and not ingest that. But the plastics that have microplastics and nanoplastics that have localized to muscle, we have to be more careful about that. Did that answer your question?
Yeah. This I'm saying, but we have to think in a different way. As the days passes, it is invisible and it's mixing with the ecosystem and getting, how should I say, infected or somewhat in everywhere actually everywhere.
Right.
Plant can absorb even, and the human body obviously from the any intake of fish or other.
And I don't know if you're familiar with this, but there's been some studies that show that cardiovascular issues occur more in people that have ingested microplastics. So people that ingest microplastics that are at higher risk for clotted blood vessels, which is scary, right? Because
Okay, please.
Yeah, one supplement just.
Very large session.
it's a discussion.
Very large. Thank you, Mr. Chairman. Actually, there is two issues. When aquatic organism engulfs a whole plastic, that kills that animal because they cannot digest. So undigested and accumulation, huge amount of plastic inside their digestibility system kills that animal. But the microplastics and enter into the muscles, that might not kill that animal immediately, but that will kill whoever consumed that fish. So that is the human. So both are detrimentals in either ways. Thank you.
So does it mean it
Because the herbivorous fish consume filter feeding systems. So then whenever the filter feeding along with the herbivorous phytoplankton, zooplankton then consume, then along with that animals or organisms, the microplastic goes to their systems. So that is also different.
And then what we see is that coating phytoplankton. So coating like algae in the ocean. So we have that.
Sir, I have a last question.
Your question.
As a perspective, we all know that plastic is a growing concern throughout the world. But interestingly, it has been detected or observed in the hair to the human being. In that case, it has been critically criticized the method of measurement or detection. How can you assure the accuracy of the detection method?
Right, so there's a lot of issues when you're working with microbiota, right? You want to make sure that you have only clean areas, you have to have hair filters. We're doing also blanks, so that means that next to the sample, we have another petri dish that has exactly the same thing with the same exposure, and we go through the compare one and the other. We only use like non-plastic materials throughout the whole process of taking the samples and like analyzing them. And we actually, so we are developing best practices and training to be deployed to other universities and also to train our students so they can go into the workforce and do that, do the like high quality microplastics detection. So yes, I mean, it's a challenge for sure, and it's not easy and it's not cheap to do it well. But I think that if you're using the same methodology, independent of across, and you're just comparing, you're going to have decent results, so long as you use, for example, a blank. So you don't have to have the very expensive, like, clean hoods that we're talking about here, and you don't have to, you know. So there's different levels of like safeguards that you can implement in your protocol to make sure that you have to take it and go out to publication. And the reviewers are not going to tell you, no, you cannot publish this because we understand, we got to start somewhere, right? And if we don't have the baseline data, we're not going to be able to see how it's evolving, right? So don't let that discourage you. As a lot of people don't want to go into microplastics because it's messy and because it's sometimes a lot of work to not get it published. But we are funded by the National Science Foundation, so everything that we produce in terms of training will be available for free throughout the world, so you guys can actually use those protocols so you can actually implement them in your own labs. Thank you.
And before that, I'd like to thank and congratulate Dr. Annabella for presenting your slides. And this was actually about the effect of temperature and pollution, but from that only the microplastic got very interested. It's very good anyway. So thank you very much. Thank you. A big hand for Annabella. Thank you.
Thank you very much. Thank you and congratulations for your nice presentation, and thank you for all participants also. And our session is in here, and second session will be start on 11:45. We will be here on 11:45 after some break. Thank you.
So I'd like to conclude with 3 words again. Thank you very much, participants. And before that, I thanked Dr. Annabella. And she presented very nicely, and I must congratulate him to finish all these things in time. And this was great, really. And it has, although that climate change is getting concerned throughout the world, but every time I'm getting confusion about this war, the climate change. It's really big, and there are a lot of concepts and a lot of issues. Still, there are a lot of hearts and many things, especially from the USA. This Paris Agreement, and it's a big funder is USA. And the then President Barack Obama, he was very curious about pouring pan on that issue and the global crisis. But Trump, with a single strike with the previous president-elect, stopped everything. And again, being elected from the same part, different party, Joe Biden, he came president-elect, again he signed as president-elect, oh, it will be high. So these are the things, it's happening for the climate change, it's a big political issue, and the Kyoto Protocol is signed all over the world, and even the poor country like us, we are getting a big amount of money for the compensation. These were utilizing in a different way. This is another question. So there is a big deal on the climate issues, and you have presented part of it. I don't know you have got money from that fund or not. So it would be very nice if you can get and do some research on it. And still throughout the world, it's becoming crazy actually, the climate change, everywhere. Anyway, still some are anthropogenic, obviously. and some are natural, we don't have any control over it. So what we have in our hand, we should be restricted. For example, plastic. Throughout the world, we're getting fancy and we're polluting the environment at the cost of everything, many things. So we should have somewhat a restraint in our expectation, because these issues are actually from the work of the human beings. They're creating it through their hands, through their feelings, through their comforts. So this way we can make keep the world, this world universe happy and livable. Getting the GDP better, we are sacrificing the peace in our life actually. So in this way we have to think, and human behavior should be changed to have a change in the climate. So let's hope for the best, and especially the participants. You participated and commented on it in a very nice way. My co-chair, and finally Dr. Anabella, you traveled a lot. You took travel to come out here, and thank you very much. And this is the end of the session.